While this video will focus on how to use IGV, we would like to note that other genomics viewers are available, such as the UCSC Genome Browser. This is a web-based application and has many published datasets and annotations available for your use. We prefer IGV for ease of use and customability, but depending on your research, you may find other viewers more suitable. IGV, or Integrative Genomics Viewer, is a Java-based application provided by the Broad Institute and is designed to visualize many forms of genomics data. As IGV is Java-based, we will use the following command line to initiate Java and load the igv.jar file. This command requires the -jar and -xmx options. -xmx designates how much memory to allocate to IGV. This specific command is allocating 5 gigabytes. You may need to allocate more memory depending on your needs. Using this command will bring up IGV in a separate window. The general layout of IGV contains horizontal tracks for loading and viewing data. This uppermost track displays where along the genome we are currently viewing. And when IGV is first loaded, all chromosomes will be displayed. To select a particular chromosome, you may click here or select from this drop-down menu. Additionally, you may type in a specific region using this box. The genome we're currently viewing is displayed here, and you may select other genome sequences by opening this drop-down menu. By default, IGV provides a number of genome sequences via the Broad Institute. You may import a genome of your choosing by going to File and selecting Import Genome, and then providing the necessary files and information. You can scroll along data by clicking on a track and moving the mouse left or right. And you can zoom in on a particular region by clicking in the upper track and highlighting that region. You may further customize zooming by using these buttons to zoom out or in. Now that you have a general understanding of how to use IGV, let's discuss the various types of data that can be visualized. IGV can load many types of files, which can be roughly divided into two categories: quantitative data, such as read pileup or coverage, and qualitative data, such as gene annotations or transcription factor binding sites. We will go over a few examples of each type of data. Quantitative data refers to coverage or read pileup along the genome, which when visualized will show how many reads are mapping to a specific location. Coverage data will look different depending on the type of sequencing performed and protocols used. For instance, RNA sequencing will show reads mapping to exons and other transcribed regions, while genome sequencing should have a uniform coverage along the genome. We will discuss 3 different file types for storing and viewing quantitative data. These are BAM, BEDGraph, and WIG files. Each of these files has a unique format, and visualization on IGV varies slightly from one another. Here we have an example of a sorted BAM file loaded onto IGV. The data is displayed into 2 separate tracks, a small, condensed coverage track above a larger track with a more zoomed-in view showing each individual read. I have selected the Color by Strand option in IGV to strand-specifically color code reads in this track. Remember, a BAM file is the binary version of a SAM file. You must load the sorted BAM file onto IGV as well as have an index of this file in the same directory. For more details on working with SAM and BAM files, please see video 1. Now we are looking at an example of a BEDGraph and WIG file loaded onto IGV. As you can see, this is quite different from that of a BAM file. These files display a condensed view of coverage very similar to one another. However, a BEDGraph is able to display strand-specific data on a single track, whereas 2 WIG files and 2 tracks are needed to do the same. This is due to differences in the formatting of each file. A BEDGraph can store 2 data points for every nucleotide in the genome, while a WIG file can only store 1 data point. Your type of data will dictate which file type is best for visualizing quantitative data. I prefer to use a bed graph for viewing my RNA-seq data. The other type of data commonly visualized on IGV is qualitative data. Examples include gene annotations, transcription factor binding sites, and replication origins. Basically, anything that qualitatively describes the genome will fall into this category. We will discuss three types of files for storing and viewing qualitative data. These are BED, GFF, and VCF files. BED and GFF files are capable of handling most every type of qualitative data, and for some projects, may be the only file types needed. As you can see, visualization on IGV is identical between a BED and GFF file. The differences in these files lie in the formatting and detail of information stored. Here I'm showing an example of both the GFF and BED files. The GFF is a tab-delimited file with 9 columns, while a BED file, though also tab-delimited, can vary between 3 and 12 columns, with each column adding some level of information about the annotation. If you will be working with GFF and BED files, it is important to know that BED files use a zero-based coordinate system, while GFF uses a one-based system. This means that nucleotide 1 is actually nucleotide 0 in a BED file. Additionally, the stop coordinate in a BED file is exclusive, meaning that the annotation stops at 1 minus the stop coordinate. Please take a moment to look at the start and stop coordinates that I have highlighted here. Though the numbers differ, they do refer to the exact same annotations. I would also like to bring to your attention the BEDTools Suite of scripts designed to carry out many useful tasks using BED files. In most cases, you can use a BED or GFF file interchangeably with these scripts, and a few will require BAM or other files. Some important BEDTools scripts to mention include, but are not limited to, the following. Intersect BED. What overlaps between two files? Merge BED. Merge together any overlapping annotations. Genome coverage BED. Convert a BAM file to a BED graph file for visualization of quantitative data. Coverage BED. Get read counts for annotations. Closest BED. Compare two files, find what is closest to an annotation. The last qualitative file type we will discuss is the VCF file, which stands for Variant Call Format. This file is used for visualization of genomic variants such as SNPs and indels. VCF files are generated by variant calling programs. You will learn more about these programs on day 6. Visualization on IGV is split into 2 tracks, a small, condensed track above a larger track showing all of the information stored in the file. In this example, we are looking at the genomic variation of 20 yeast strains relative to a reference genome. Each color block in this region marks the presence of a variant, while the upper track acts as a summary, marking the presence of any and all variants at that genomic position. Please note that you must have an index of a VCF file before loading onto IGV. Indexing can be performed from the command line using the command IGV-tools and index. Alternatively, you can do this via IGV by going to File and selecting Run IGV Tools. Now that you are familiar with the basics of IGV and the various file types that can be used with it, let's take a more in-depth look at how to use and customize tracks in IGV. For this example, I have loaded a BED graph file along with gene annotations from a GFF file. File names for each track will be displayed in this region here. Right-clicking on a track will bring up various options for customization. For instance, we can rename this track, like so. When annotations are first loaded into IGV, they will be displayed in a collapsed form. To change this, right-click and select Expanded or Squished to view all the annotations in this region. After zooming in, we can see that the track height for the BED graph is rather small. To change this,
select Change Track Height. Now we can see more detail in our coverage data. We can also see that the bed graph is strand specific, such that coverage from the plus strand of DNA is in the positive range, and coverage from the minus strand of DNA is in the negative range. This can be color-coded by selecting Change Track Color by either positive or negative values, and selecting the desired color. Now we have plus strand coverage in red and minus strand coverage in blue. The scale of this track is displayed here and can be customized by selecting Set Data Range. Likewise, we can change the color of our annotation track by selecting Change Track Color. To load additional files, go to File and select Load from File. Once you have all of your tracks loaded and customized to suit your needs, you can save this session for future use by going to File and selecting Save Session. This way, the next time you open IGB, you can go to File and click Open Session, and it will load all of your tracks along with the formatting that you had saved it with. You may also find that you would like to take a screenshot of IGB for use in a talk or for your lab notebook. To do this, go to File and select Save Image. This will take a screenshot of the current view of IGB in the form of a PNG. Again, right-clicking on a track provides you with many options for formatting that track. We have only touched on a few of these here. When viewing your data, take some time to explore and play with these options to best suit your needs.